Hi, I'm here today to show you meiosis and simulate it with some fake chromosomes. So take a look down here at my cell. I have a nuclear membrane right here that houses my chromosomes. I'm starting again with the diploid number of 4, so I have 4 chromosomes inside of my cell. And then outside here is my cell membrane, and I have some organelles as well, like mitochondria and Golgi apparatus and vacuoles and all that fun stuff. So to start meiosis, we do the same thing that happens in mitosis, where in S phase, all of your chromosomes are copied. So I'm going to show all of our copies of chromosomes coming in. At the end of S phase, even between or before meiosis begins, you have a potential of 8 chromosomes in this particular cell. So let's start at the top. Remember, meiosis is going to go through 2 rounds, not just 1 round of division. And so I'm going to call things prophase 1 for round 1 or prophase 2 for round 2. So let's go through prophase 1. In prophase 1, something really unique happens. So first, very similarly to mitosis, all of your sisters find each other, all your identical copies, right, find each other, and they will hook up at that centromere region just like we saw in meiosis. But this time something interesting happens. Not only do the sisters find each other, the identical copies, but we also find our homologous partners. So remember, your dad will donate 1 set of your homologous chromosomes and your mom the other set. Those have all been copied. You can see that we have the same sequence of genes on these chromosomes, but they have different alleles or different forms of the gene. So then what happens in crossing over, where the chromosomes are kind of laying on top of each other, enzymes run along the chromosomes, and they actually, I'm going to use a little Lego device, but they're going to actually come along and they're going to switch equal lengths of your homologous chromosomes. This is a process called crossing over. And so enzymes will run the length of your chromosomes, and then they will switch out equal pieces of one chromosome to the next. When crossing over is finished, then prophase 1 ends for meiosis. We move to prometaphase. This is where your nuclear membrane will disappear. So nuclear membrane disappears temporarily. And this time your tetrads hook up to your mitotic spindle or your spindle of microtubules. The microtubules will grow out and one will attach to one set of homologous chromosomes, and then one tubule will attach to the other set. Now, once you're done with prometaphase and you're all hooked up, all the chromosomes are moved until they're in a line in the middle of the cell, and that is metaphase 1. This time, notice, instead of having sisters along the line, we have tetrad sets, or all 4 homologous chromosomes in a set in the middle of the cell. You still run a checkpoint, and then at anaphase, the tetrad sets separate, right? Telophase 1, your chromosomes are going to get pulled to the opposite ends of the cell. In telophase 1, your nuclear membrane will sort of return. Oftentimes at this point, your chromosomes kind of relax a little bit, so they'll kind of unhook from each other and unwind, check themselves, etc. Cytokinesis 1, the rest of the cell contents splits. Now, we will start meiosis 2. So in meiosis 2 is a lot like mitosis. So in this phase, we're going to start with these 2 cells, and all of your chromosome sets will re-hook up in prophase 2, so that we have these like chromosome sets. In prometaphase 2, your nuclear membranes disappear. In metaphase 2, your chromosome sets now line up in the middle of the cell. In anaphase 2, the chromosome sets separate and are pulled to opposite ends of the cell. And in telophase 2, your nuclear membranes will return. So each of these sets of cells will get a nuclear membrane to return around it. And then once the nucleuses have returned, then your cytokinesis 2 will occur, or cytokinesis will occur to split your organelles and your cells evenly, so that you get equal amounts of organelles going in both directions. Now, at the beginning of meiosis, we started with 1 diploid cell that had 4 chromosomes in it. By the end of meiosis 2, you have 1, 2, 3, 4 cells, each with 2 chromosomes inside. So now they're considered haploid cells. And if you look, All of our sets of chromosomes have different collections of genes, so they are also considered new, unique cells. Thanks for watching this meiosis simulation. See you again next time.